All right. Stuart, if you want to go ahead and start sharing your screen.
Yeah, sure. Is it all right?
Yep. All right. We're good to go. Hi, everyone. Welcome to our next webinar in Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. This webinar is brought to you by Plantae, the online open community for plant biologists, powered by ASPB. Next slide, Surya. I would like to give a special thank you to all our ASP members who are attending today. Your ASP membership dues help support making the webinar possible. For any of you who have not yet joined ASPB, you can join today and use the discount code %10 to receive a 10% off discount on your registration. ASPB members get early access to this webinars, and you can learn more about ASPB and opportunities we provide at aspb.org. Next slide. In addition, please be sure to check out our YouTube channel where you can find many other exciting talks. This talk is recorded and will be shared on our Plantae YouTube channel, and you could rewatch or reshare with any of your colleagues. Next slide. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most out of your experience today by attending this webinar. If you are experiencing technical issues, please let us know about those by using the chat box or by emailing me at jpadilla@aspb.org. If you have questions for our guest speakers, please add them to the Zoom Q&A section. Our moderator will be sharing them with our presenters. If you have trouble connecting or need to leave the webinar early, again, know that this webinar is recorded and will be made available on our YouTube channel. So please be sure to subscribe to our YouTube channel to receive notification when new content is posted. Well, all right, without further ado, now I'm going to turn over the floor to our moderators and speakers to share today's content. Take it away, everyone.
Thank you very much, Jason. Good morning, good afternoon, good evening to everyone who are attending from various parts of the world. My name is Saurav, and I am one of the Plantae Fellows of 2023 cohort. Welcome to Plantae monthly webinar on how to create widely accessible and easily digestible research outputs, featuring Professor Detlef Weigel and Dr. Arunabha Bose as the invited speaker for the webinar. The session will be moderated by 3 Plantae Fellows, that is me, Abdul Kabir, and Maria. Here is the outline of today's webinar. Speaker introduction followed by a quick moderator introductions, and then we'll jump into our first talk of the webinar, followed by a second talk. Each talk will be roughly 15 minutes long. After that, we'll have a question and answer session, and then the concluding remarks to draw the curtain. Please feel free to drop in the questions in the chat box during the talk so that those questions can be answered during the Q&A session. Okay, so without further delay, let's get started. It's my pleasure to introduce the speakers of the webinar. The first speaker of the webinar is Professor Detlef Weigel. Professor Weigel is a German-American scientist, is currently a director at the Max Planck Institute for Biology, Tübingen. He is a member of the US National Academy of Sciences and the Royal Society, and a recipient of many scientific awards, such as the recent Novozymes Prize. Professor Weigel has an extensive record of service to the scientific community, having served on a series of editorial and advisory boards, and he is a strong proponent of open science. He sees as his most lasting contribution to science the outstanding doctoral and postdoctoral researchers, as well as undergrads and research assistants who have worked in his lab over the past 3 decades. Welcome, Professor Weigel.
Hello.
And the second speaker of the day is Dr. Arunabha Bose. Dr. Bose completed PhD in biochemistry from ACTREC, India. After that, she went on to conduct postdoctoral research studying chromosome biology and cell division at the IGC Lisbon. She has been a scientific editor at Data in Brief and Method X since July 2021 and March 2022, respectively. She is passionate about simplifying science and making it accessible. In her free time, she likes tasting recipes, trying different coffees, and going for long walks. Welcome, Dr. Bose. All right, now I am going to turn it over to one of my co-moderators of this session, Maria. Over to you.
Thank you, Surab. Hello, everyone. It's my pleasure to introduce you to today's moderators. First, I would like to introduce Abdul Kabir. He is interested in molecular plant pathogen interactions and population genetics. He has received a bachelor degree in plant biology from Nigeria. And during this time, he engaged several student communities within and outside the university walls on the importance of cross-cultural understanding, diversity, inclusion, and equity in the pursuit of research and academic excellence. He has a passion for scientific writing and food security advocacy campaigns. In his free times, he enjoys writing, therefore Plantae Fellow, discussing science, playing soccer, and enjoys nature. The next moderator that I have the pleasure to introduce is Surab, who you have just met. He is pursuing his PhD in genetics at Monash University, Australia. His studies involves understanding the molecular mechanism of temperature responses in Arabidopsis thaliana. Apart from research, he enjoys very much playing cricket in his spare time. And lastly, I would like to take the time and introduce myself. My name is Maria. I have a PhD in plant pathology from the University of Amsterdam in the Netherlands, and I am currently the open access officer at Utrecht University, also in the Netherlands. And in my spare time, I enjoy writing about plant science stories in my own blog, The Green Leaflet. And without any further ado, I would like to give the floor to our first speaker, Dr. Vego.
All right, thank you very much. Let me share my screen. Here we go. All right, so I'm really pleased to be part of this series here today. Thank you so much for inviting me. And indeed, all of us enjoy science, are enthusiastic about lab science. But as important are all the other things that we do, from educating our colleagues and people we work with to going beyond the lab. So I see that as a very important part of my job. And one of my missions I see as making sure that that is being acknowledged. It's one of the as sometimes not so great things when I see evaluations that still the publications and grants are the only thing that is being considered. These sort of things that we are discussing today, in my view, are as important a part of being a scientist, being an excellent scientist, really. So I want to give you 3 examples where producing easily digestible and simple to understand research outputs have been important in what I do myself. And we go from far away from the lab towards getting closer to the lab with these 3 examples. So the first example will come from what is called the Children's University. Here in Tübingen is where this started. This is now all over Germany, where researchers give lectures in a university-style setting, but it's geared to children at different ages. Then I'll discuss a little bit what my approach has been when I give public lectures on genome editing. And then in the end, some of you might know that I'm quite active in social media, especially on Twitter, and where I think Twitter threads is really an outstanding way to communicate your own science and really forces you to abstract and simplify things. So first example, Children's University, there I talk about evolution, and the title of my presentation is why the shepherd dog is a shepherd dog and not a duckle. And so you can already see what I take there as an example. While I myself work on plants, I try to make it general. And what I think where it starts with is that we don't shy away from using everyday language as appropriate. So I present diversity of Arabidopsis, but instead of saying, phenotypic diversity, I just point out that some are small, some are large, others are short, others are tall. And of course, a slight digression here. Again, many of you have seen this, but these are not things that I invented or came up with, but this is often considered the Hemingway style of writing. This is a phrase that Hemingway didn't coin, but really fits here well, that clear writing, and one could also say clear presentation, comes from clear thinking. So you should be sure what you write about, you should be sure what you present. And once you're sure about this, then you can actually do it in simple words. So often when we use difficult phrases, it's ourselves who have difficulty understanding it. And it comes from us ourselves really not being quite certain what we want to express. And very important also in scientific writing and presentation to not be afraid of short, concise sentences. So I think it's perfectly fine in a scientific paper to have phrases like these. Was this indeed the case? No, it was not. So many people would shy away from this, but this would not look out of place in a Hemingway novel. And if it doesn't look out of place in a Hemingway novel, it should not look out of place in scientific writing. Along these lines is to avoid unnecessary embellishments, unnecessary adjectives and adverbs here, a typical thing. We observed a dramatic increase. Dramatic is already dramatic, so you don't have to say that it's very dramatic. Either it's dramatic or it's not. And then finally, to really focus on the essential details. And so these are all things that I apply in my writing, but I also consider when I go out there and talk to children. In writing and especially in science communication, I think it's very important to think of analogies or metaphors that your audience can relate to, because this allows them to abstract. So I'm a plant biologist. I talk about diversity of plants. I talk about plant evolution, but I don't want my audience to go home and say, well, today we learned about the evolution of plants. I want them to learn general principles. So one of the things that I came up with many years ago was actually, and I was inspired by visiting a local museum from the Mercedes-Benz factory on the history of automobiles, that I use automobiles to bring home this idea of how different variants evolve from a common ancestor, and how this common ancestor could look very different. And you can easily imagine how this provides many opportunities to talk about all kinds of aspects of evolution. Similarly, I like to employ images that allow for emotional responses. So talking about evolution of dogs and wolves, obviously, when kids see this, they will immediately respond to this. And as scientists, we often think that emotion has no place in what we do, but I think we're really kidding ourselves if that's what we are trying to do, to take emotion out of everything we do. We are humans, and so I would say let's embrace it rather than trying to avoid it. What as scientists, we also often forget these sort of graphs here, for example, they are something that we immediately understand. So we look at the number of years there, and we understand that this goes from left to right, and that there is this increase. But this is because since we were undergrads, we've been seeing these graphs, but when you talk to people out there who don't see these graphs every day, they might find it more difficult to understand. So here I employed two tricks. So I use these rice grains to make clear that this is about the rice itself. And then instead of showing an static graph, if you use animation like this, it becomes much clearer what is happening here, that something is continuing over time and then also increasing over time. All right, so let's switch to this second example, or the second set of examples that I want to give. And these are examples from public lectures that I give on genome editing. And this, again, it relates to how I see my task as a scientist who is being paid by tax money. This is obviously not something that my employer demands. But as a scientist, I feel strongly that we all have an obligation to go out of the lab and communicate both in terms of teaching others, but also standing in for what we think is right. And so there, what I found is that approaching a topic in an indirect manner. So when I talk about genome editing or transgenic plants, I talk about genetic diversity. And of course, I could just start and say, well, here's a plant and make a tiny change. But that's really difficult for many to imagine or understand what this is really about. So starting with images like these, where I compare teosinte and maize to drive home this point that plant breeding has caused a dramatic change, then later allows me to talk about much more subtle changes that we create by genome editing. And in this context, then, for example, I will talk about mutations that we find between different varieties of corn, where the number of genes between different varieties can differ greatly. And again, say, what does this have to do with genome editing? Again, it's this approach of getting to the topic in indirect manners, in a way preparing the audience for what in the end I want to discuss, that these sort of changes that we create by genome editing are really small compared to what is already out there in the natural world. And again, using simple images to convey profound messages. So I found this to be extremely powerful when I talk to the audience afterwards. So I could say, you know, these mutation rates that we have measured, and in every plant, there are so and so many mutations, but that is extremely abstract. So this very simple visual where I show this field of cereal, and then just to tell the audience, look, here is this field of cereal. And normally when you go out there, you look at this field and, well, you think all these plants are the same. But actually, these are all mutant plants, and they're all different from each other because they are spontaneous mutations. This really gets people thinking, because you challenge them to change the way they look at the world. And I think there's also another important lesson there, not to underestimate your audience. And so this is one of my sort of favorite devices. One shouldn't be afraid of stating the seemingly obvious. So again, this is with genome editing. It's something that I often start with when I give presentations on genome editing, where I show these DNA sequences. So I obviously explain this, that these are DNA sequences, that we have the 4 bases, A, T, G, and C. And then I ask the audience whether they see differences between the top and the middle, and they see differences between the middle and the bottom. And then I explain to them that according to the legal definition in Europe, there is a difference between the 2 DNAs in the middle and the bottom, because if one is a spontaneous mutant and the other one is an edited mutant. But there's no difference between the top and the middle, because the top is a wild type sequence and the middle is a spontaneous mutant. So again, very, very simple devices, but very, very tangible, rather than just stating numbers. All right, so I want to conclude with Twitter threads, which, as I said, I found really an excellent way of communicating science, and give you an example from my own, from our own work. This was a preprint that we put out there a few years ago on mutation biases, and then used a series of tweets, what is called a Twitter thread, to introduce the findings of this manuscript to the Twitter audience. And so the way this works is you formulate a tweet, and then you reply to your own tweet, and automatically it forms this thread one after another. And I really like this medium because, again, it forces you to focus on the essentials. So the figures in the paper have typically several panels, are quite complex. You have legends that run to sometimes 100, 200 words or so. And so being forced to express this in 280 characters really makes you think about what is the essence of the work. And I don't want to read this all out to you, but you can see, I think, again, that this is all relatively, these are simple sentences. So, for example, could mutation rate bias be adaptive? So I think you all can think of ways that one could express this in very fancy language with many, many more words. But this simple statement, just 6 words, captures what's in the paper really, really well. So then you just add to these, and one of the things that I like there with Twitter, that you can inject some levity into science communication. So, and again, so here you see how I, follow this advice that I gave earlier, this Hemingway style of writing, and that everything has to be short. So here is a relatively long, relatively complex sentence, but then this very short statement afterwards. And I think you can all see how this is very powerful. So we start with this, very short statement, the acid test, colon, then this relatively long sentences here. Do we see effects at gene level indicative of evolutionary constraint predicting mutation rates? And then we do as the answer. So again, you can think of, one could express this in many more words, but to make this very simple statement, we do, is extremely powerful. And then over on the other side, then, as I said, inject some levity, because we are all humans. We are not robots doing science, and it's perfectly fine to communicate that to the greater world, that we have emotions also when it comes to our own discoveries. All right, and that's really all I wanted to share. And I'd be really happy if there are questions that I can answer later on. Thank you.
Thank you very much for a very clear and nice presentation, I would like to add. And now I would like to give the floor to Dr. Bosset, and we will take the questions, as Surav mentioned, at the end of the presentation. So do not wait and start writing them now.
Okay, thank you, Maria. Hello, everyone. Thank you for having me here today. I think ever since I've heard about this webinars, I've been going back and looking at the previous webinars. And I think the range of topics that you guys have covered is amazing. So I'm very happy to be here. Thank you. Just give me a second. Let me share my screen. Let me know when you can see my slides. I think you can see it now.
We can see it now. Yeah.
Good. Okay. So today I'm going to talk to you a little bit about how you can make your research more accessible. And just to give you a brief overview of what I'll be discussing today, I'll be introducing you to something called research elements and data in brief. We'll be going over how you can actually submit your research to data in brief. And we'll also talk about why data repositories are important, what they are, and how you can use them. And finally, I'll end with a few links that you can find useful, and also let you know how you can join us as a reviewer. So to start with an introduction to research elements and data in brief, if we can, I'd like to start with just imagining how you start, if you were to start a research project. What you do is first you do an entire literature survey, you do a review, you see what topic you actually want to focus on, and then you build a hypothesis based on that review that you've done. Once you have a hypothesis, you know what methods you need to do to achieve your results, the experiment you want to do. Then you actually perform the experiment, you collect a lot of data, and then you analyze that data using different softwares, different machines that we have. And then finally, once everything is done, you compile all of this into a results, into a research article, and then you communicate it via a research article. Now, all these steps are actually different kinds of research outputs or research objects. And via research element journals, you can actually get credit and get citations for each part of this entire cycle. So what I mean is you can have something called as a data article, which is what we do at Data in Brief. What we have is data articles. These focus on the data, on just description of the raw data that you've collected. You can also have a paper describing the software. So if, for instance, you've used a particular code, if you've generated some code to help you analyze your results, whatever they may be, you can communicate, you can get a citation for that code in a journal called Software X, which is what we have. You can also receive a citation for the methods. So you write a methods article, which focuses entirely on the methods that you've used. So you write this in a way that makes it reproducible, that helps others find your data, others find the methods that you've used, and you also get citations for all the work that you've put in. That is what research elements journals do. And then going into the journal that I work with, which is Data in Brief. What Data in Brief is it's a multidisciplinary open access peer-reviewed journal, which publishes short data descriptor articles that describe and provide access to research data. And this contributes to data sharing and reproducibility. The way that this happens is, what we require is that you upload your data onto a repository, and then you write a data article describing the data. So say, for instance, you had, for instance, sequencing data, which you would usually upload on, say, GenBank or NCBI. So via a data article, what you do is you describe that raw data, just the data. It does not need results. It does not need conclusions or a discussion. You just describe the data, you describe the methods, and you describe how you've collected the data. And in this way, it contributes to data sharing and reproducibility. So what is in scope for our journal? We welcome raw data, which is, like I said, sequencing data, or say, for instance, if you've conducted a survey. So the actual questionnaires and the actual responses that you collect during that survey, that would be raw data. If, for instance, you've done mass spec analysis, the actual mass spec data files is raw data. So that is within scope for us. What's also within scope is simulated data with the related code, of course. Negative data, this is something I feel quite strongly about, which is, say that you have a hypothesis, but your results don't really match your hypothesis. So how would you go about publishing that? Because then the research article that you write would be a little difficult to write. Whereas if you write a data article, you're just sharing the data independent of the context, independent of the hypothesis. And this helps other people find that data, and it saves time and effort that other people would put into replicating that same thing. So they know that this is the data you've collected and that is how it works. That is also within scope. And like I said, survey data. And of course, we have different policies for sharing sensitive data, which you can find on our journal homepage. We have policies for secondary data and for reviews. What we're not looking for, or what is not within scope for our journal, is supplementary files from a review article or data from case report studies. And let me just walk you through how you can go through submitting a data article or submitting a manuscript to our journal. It's a simple process. It's a 3-step process. What you do first is you upload your data into a repository. I'll talk a little bit about repositories later. We have a webinar on repositories, an education webinar that can help you identify the correct repository. You upload your data there, then you go on to fill in our submission template, which you can find, there are links to all through the presentation. And then once you've completed the submission template, you submit your template into the online submission system. And we also have a webinar to help walk through this entire process. And once all this is done, once you've submitted your data, once you've submitted your data article, your article will undergo peer review and hopefully get accepted. And upon acceptance, I've just added a few screenshots of a few articles that we've had to give you an idea of the different kinds of articles we publish. And all of these are clickable links. So please feel free to go through any of this if you find it interesting. I've also added a link to other full-length articles that we have. So now that I've spoken to you about data in brief and told you that we insist on data being deposited in repositories, let me just tell you a little bit about repositories, why they're important and how you can use them. Now, data repositories actually, just as is in the name, they store data. So they store data sets. These are databases to store and share scientific data. Data shared in an appropriate repository will have a unique digital object identifier, or a DOI, which is assigned to them, making them findable and often accessible. And what they will also have is version control. So what this means is data sets may be updated. You can go back and edit your data sets, but what they will contain is they will contain a record of each version. So as you edit your data set, it will still have a version 1 or version 2. So if anyone needs to go and find it, they can find each of these versions as the data undergoes more maturity, or as you add more samples to your data set. So like I said, these new versions will also be recorded, ensuring that previous versions remain accessible while the data sets may be amended. And uploading data onto a repository and sharing the DOI is mandatory for publication at Data in Brief. And why this is useful, or how this is useful to everyone. This is actually useful to everyone in the cycle, because for authors, this improves data communication, confidence, and transparency. Because if usually what you do is you will communicate, or what I have done previously is I've written research articles, and sometimes you just have your data in the supplementary files, which usually no one reads, and it gets lost, and it's your effort, and you want it to be findable. You want to get credit for that too. So if you share your raw data and you link to your raw data, anyone who goes through your research article can find it. And then they can perform their own analysis into that data while giving you credit for what you've done. And this improves confidence in your research, and it also improves transparency of research. And like I said, it improves the findability of your data set via the DOI linking. And it improves scientific progress by helping other researchers. So it decreases the time and effort that would go into otherwise replicating the same thing that you've already done. So you get credit for what you've done, and people can build on what you've done while giving you that credit. For reviewers, this helps because it improves review quality, because they can go through your actual raw data, see what you've done, see how you've done it, see the methods you've used, and this leads to better quality manuscripts in the end. And for readers, it facilitates reproducibility and replicability, like I said before, and it also helps in conducting reviews. So if someone wants to write a review on something, on a topic of their interest, if they find a data set that there already exists on something that they are interested in, you get credit for that too. You get credit for your work, and you get someone else using your data set while citing the same. This is how it improves and gives value to everyone. And there are thousands of repositories actually where you can upload your raw data. So it's important to be able to identify the correct repository. And you can find a registry of these repositories, which can be used to identify appropriate repositories. There is a link here. But what you need to consider in this case is the subject area. So for instance, if I have a data from earth sciences, I would usually discuss this with someone in my lab or with my PI and see what sort of repositories they are used to using. So if I have something in the earth sciences, I would upload it in something called Pangea, for example. or I would upload it. I would not, for instance, upload it in a repository that handles nucleotide sequences or GenBank data, because that is not applicable in that case. So that is something you need to consider. You also need to consider sharing capabilities. So what this means is whether you have any access control, whether you are able to control who is able to view your data. And we do have this at Data in Brief also. If, for instance, you're sharing, say, census data, which is critical, or you're sharing census data from an endangered species, you don't want that to be immediately accessible to everyone. Or you're sharing survey data that is critical, you don't want that to be immediately accessible. So what we do in that case is you hide it behind a safe wall or an embargo. And then you can also have a data usage agreement. You can find all of this on our journal homepage, but that is also something that you need to consider. The other thing that needs to happen is you should consider whether there are permanent links. This is important because you want that link to be present forever. It shouldn't happen that 2 years down the line that link gets taken away and then no one has access to the data. So that is also something you need to consider. I think nowadays there are a lot of universities that have their own repositories, and those are good as long as they provide you with a stable DOI or a stable link. You also need to consider storage limits and, like I said, time span before, the reputation in the community, and version. So versioning was, like I said, version 1, version 2, as the data set evolves. What we do is we strongly recommend the use of a specialized repository, such as, for instance, GenBank for sequencing data, or Pangea for earth sciences data. Or say, for instance, you have mass spec, you would upload it on say, Proteome Exchange or something. And the reason this is important is because all of these repositories, most of these repositories, they also do their own quality control before making the data visible and available to everyone. So that acts as an extra badge of confidence in your data. When you say that you have uploaded it to GenBank, you know that it has undergone some level of quality checks, and that gives your readers or your reviewers some extra confidence in your research. And that improves reproducibility and findability, as I said before. So that is what I had for today. And now I'm just going to, if you'd like to become a reviewer for us, for Elsevier, for Data in Brief, what you can do is, if you submit a data article to the journal, you'd be automatically added to our reviewer database. And you can also create your account and then sign up as a reviewer using this QR code. It works, I've checked, so please feel free to use it. Yeah, that's about it. In conclusion, what we do at Data in Brief is we publish data descriptor articles. These describe data sets stored in repositories, and publishing your data in Data in Brief makes you and your research more visible and citable. And just to add one small thing, we're getting an impact factor this year in July 2023. We don't know what it will be exactly, but yeah, we're very excited about that. So let's see how that goes. And this is, I've just added a bunch of links that I thought you might find useful. So please feel free to go through this. And that's all I had. So thank you so much for your time. Please, I look forward to any questions you might have. Thank you.
Thank you very much, Dr. Bose, for introducing data articles to our audience. I would like to thank both speakers, and we have already received many questions. And I will try in order, and let's see how many we can answer live. The first one is for Dr. Weigel from Michael. He congratulates you for an amazing presentation. And he wonders how many tweets in a thread is considered optimal for announcing our observations in Twitter. So do you have a Twitter number that you recommend?
Yeah, I would say a dozen, half a dozen to a dozen, or 5 to 15. So I would try to, have at least one tweet for each figure. So not almost certainly not the entire figure, but taking the most important panel from that figure. Obviously, when it gets too long, then you lose that it loses impact, because the idea is really to announce briefly and then to entice people to read it in full.
Thank you. For Dr. Bosset, what is the difference between data in brief and basically a repository such as proteomics and RNA-seq data? So what is the added value of article?
Yeah, that's a very good question. So the difference is that if you look at something like, if you have, sorry, I forgot what was the repository you mentioned, Maria, but the difference between a repository, so say Proteome Exchange or something like that. The difference between something like that and data in brief is that Proteome Exchange is a repository. So that is somewhere you actually just store the data, whereas data in brief is a journal. So what we do is we need data to be stored on a repository, and then using a data article or using data in brief, you can actually describe that data. That's what the difference is. So we encourage sharing data on Proteome Exchange and then writing an article about the data. That's the difference. And the added value it has is it's an article, it's a citation that you have. So yeah, that's the difference.
Do you have any evidence that the raw data published is being used in other studies? And what would be the reason that an author would prefer to do a raw data publication instead of waiting for the full analysis of the data?
Yes, so there are two questions there. I'll start with the first. Yeah, we do see now that this is being reused, because we've actually started a new article type recently called Letters to the Editor, because we noticed that there were some people who had performed experiments using the data that had been published at the journal, and they had some comments they wanted to make about the data being used, about the data that they had used. So yes, we do see it being used. We do see a lot of, there are citations. And like I said, now, if we get an impact factor, when we get an impact factor, we'll probably see an increase in the number of citations also. And the second question, why is it useful for you to publish your data? Well, what it does is, briefly first, is when you upload your data onto a repository, like I said, you have a stable version of it. So it doesn't really matter if you write a data article at, it's not important at which stage of the research cycle you write your data article. So it's not really important that you write it in the beginning when you just started collecting data, or at the end. What I would recommend is you write the data article once your data set is complete, once you have a significant amount of samples, so that the data can be reused by other people, because that's one of the things that we insist on, that the data should be reusable and reproducible. For it to be reusable, it has to have a significant number of samples or a significant amount of quality control to it. So in that case, I would recommend you write a data article at the end of the research. But a data article is in no way a sort of alternative to a research article. In fact, it's a complement to it, because what we have is we also have links between research articles and data articles. So what we have is, several times we have, in fact, half of our submissions are that way, that we have authors writing a research article, and then in support of that, they write a data article, which describes the data. So like I said, it doesn't have a conclusion or a results, but it just gives you, a citation that you have peer reviewed, and it helps people who want to actually just read your data for what it is, or analyze your data for what it is. That is the difference.
Thank you, Ava. Okay, I have a very, very long question, but let's see if I can translate it. I think it's for you, Dr. Weigel. In communicating science and scientific publications, what do you consider the other strategies to engage audience beyond using simpler words? And what do you consider as the biggest challenge with communicating scientific findings?
Well, I think whenever you give presentations, the most important thing is really to engage with the audience. So the first thing would actually to look at the audience and see how they respond to it, whether they react to what you say at all, and then if necessary, based on that, to adjust what you do. There are a few other tricks, obviously, when you give presentations, so that you use humor, but this is something that you have to be careful. It has to be appropriate for the audience. So if in doubt, it's better to not try to be funny, because you might end up offending the audience. An important trick when giving presentations is to write down the first 1 or 2 sentences. So, even I, having given hundreds of seminars, I'm still nervous when I have to speak. So having that first 1 or 2 sentences really helps you, because then you're not gonna stumble, and you set the tone. I hope these are some of the answers that could be helpful.
Yeah, definitely. At least for me, it's comforting to hear that even after so much experience, just writing and making sure you're not blank at the beginning, I think that's very useful. Okay, we have another question for data in brief. It has become very popular. Can you submit a data in brief article specifically for plant traits data that have not been published? So I guess it's really data that has been collected in the laboratory. Would that be suitable within scope for data in brief?
In one word, yes. That's, if I can keep it that way. In one word, yes, it's suitable. The only thing that's important is that you upload it to a suitable repository and write the methods in as detailed a way as you can. But yeah, yes, by all means, please.
And will data in brief do any sort of checks to this type of data, or consider something?
Yeah, so all articles submitted to data in brief undergo peer review. So what we do is we have a wide editorial board, and after it comes to us as scientific editors, we assign it to someone who is an expert in the field. So for instance, if you have plant data, it would go to someone who's a scientist, who is a plant scientist or a plant biologist, and they would, of course, analyze the data and the way the article is written, and then in the end, also invite external peer review if needed. So yeah, those are the checks it goes through.
And can you republish data? So I guess it has been published already in an article. Would that data be suitable for data in brief?
If it's already published, I imagine that the data is in the supplementary files or something like that, because if it's actually raw data, I imagine it would be in the supplementary files. In that case, what I would suggest is, if possible, contact the journal that it's already been published at, and ask them if you can remove it from there and add it to an online repository, because then you can link to that online repository at data in brief, because otherwise it's duplication of data, which is not valid. So if it can be removed from there and published into a repository, then that's fine. But if not, then probably would not be within scope.
Thank you. For Professor Weigel, we have a question specifically for mixed audiences. So how do you choose the right level to make sure that it's not oversimplified, that you will bore some of the listeners, but also not too high level?
I think it's almost impossible to oversimplify things. So I would always err on the side of simplifying things. So also what I typed in the chat just, I mean, the sophisticated audience, hopefully they can actually learn, they can also learn from you and see how one can present something in a simplified manner. Now, if it's a more scientific talk, I have a little tip there that might sound not entirely serious, but I think it's actually pretty good advice, and that's the general guidelines that I follow. So when I give a seminar, I really try to keep almost everything fairly simple, that even if somebody is half asleep will understand it. And then I have 1 or 2 slides in there where there's something sophisticated. So, you know, that for those who need it, they do appreciate that obviously you are capable of doing sophisticated things. But that's just more there as sort of an advertisement to remind people, yes, it's all presented here in a simple manner, but obviously in the lab we do approach it in a more sophisticated fashion.
Yeah, very nice. So keeping few little nuggets of more intense research and so on. Yeah. So I have a question from a PhD student, and I think this question actually I would like both speakers to answer. And it's, what should I aim for publishing a paper? Should I choose an open access journal or not an open access journal? And I'm just curious to hear both views on this.
Well, I mean, I'm if I can start out there, just because I have a long history in terms of advocating for open science and open access, and I know that Anurava, of course, she told us about the advantages also of publishing in open access venue. So I see there's essentially no reason not to publish open access, except of course for the cost, and that's something that of course often weighs on authors' minds. But almost all serious journals, they are obviously predatory journals, which I'd exclude from this, but serious journals. And Renaba, maybe you can tell us whether that's true for all of the Elsevier journals, but I think serious journals, if you don't have the financial means, you can ask for a waiver. And I know, for example, at eLife, where I've been a deputy editor for well over 10 years, that I think we've never had the case that we have not granted a request for a waiver, and the requests for waivers are actually relatively small. That in itself is sort of a problem, because it is quite clear that it seems that somewhat a stigma is still attached with this, that some people who would be granted a waiver, that they still feel uneasy asking for a waiver. Is very difficult to give advice there how to overcome this, but really I think there is no editor in whose mind they would think any less of you as an author if you ask for a waiver. We all understand that the world is complicated, and what works for one author will not work for another author.
I agree.
Is that about right?
No, I agree completely with you. We are an open access journal, and I understand that the only limitation in publishing open access is the fees, the article processing charges. But like you said, Dr. Weigel, I think if there's, for any reason you're unable to pay the fees, which is completely understandable, please write to the journal, whichever journal it may be. If they're not able to give you a response immediately, they'll at least be able to direct you to the person who can give you a response. So yeah, I agree.
Thank you. So a question for Dr. Weigel, and that is regarding Twitter algorithm. So here in audience, you have probably a very well curated audience in space of academics and people interested in science. However, with Twitter algorithms favoring popular and viral content, how do you use Twitter to communicate your findings and threads to non-scientists and the public who doesn't follow you?
Yes, so unfortunately, as scientists, we are often at a disadvantage when we discuss with people who are more ideologically motivated, because as a scientist, no matter how outlandish the hypothesis that somebody else presents, as a scientist, you always have to admit, even if the probability is very small, you cannot, there are very few things that we can rule out 100%. As we know from Karl Popper, we can't even guarantee that the sun is going to go up tomorrow, even though it has been going up for billions of years. So as scientists, we are at a disadvantage there. But I would say, as everywhere else in life, being honest, I think, goes a very long ways. That where you yourself are uncertain, that you share this, and to really try to make clear that you are not driven by ideology. So when I talk about genome editing and genetically modified plants, the first thing that I always state was, I start with two things. So I say, I state that, I do consult for a breeding company so that people know that, this could be conceived as a conflict of interest. But more importantly is, we're not going to all die of hunger if we don't use genetic transformation or genome editing. But it's just going to be a lot more difficult to feed everybody in the world. It's going to be a lot more difficult to combat climate change, but it's not the end of the world. And at the end, it is all about choices. And we as a community collectively have to decide what is important to us. But I think if you admit that you are a human being, coming back sort of full circle what I said earlier, we are not robots, we are humans. I think if you admit that you are a human being and show vulnerability, I think that goes a long way. There's this overused word of authenticity, and it's not something that one should play, but it's part of being authentic to admit when you don't know things and when you are unsure, because in the end, we also want to share with the world why we are scientists, the scientific method, and that's very much part of being a scientist.
Thank you. And I think we have only 3 minutes left. So as a last question, I will take, I actually want two, but let's see. The first one is, how do you get out of the echo chamber regarding GMO to address people who are not open towards the technology? Maybe a short answer.
Yes.
Possible.
So again, so the important thing is, you can't just teach people and say, I know this as a scientist. You have to help other people to discover the truth for themselves. So really try to make them think about. So if somebody says it's like this, then ask them, why do you think it is like this? And don't just say, no, it's just different. I know that. So not a prescriptive measure, just educating the public. I think that's the message. Yeah, and taking the other person seriously. Yeah. And lastly, for you, Abba, we have one question regarding FAIR data. And is this something for the audience who is not aware, is basically checking on the data? Do you check this data in brief? And is that
yes. So one of there are 4 principles: findability, accessibility. I can't remember what the I and R stand for, but what this basically means is that you want your data to be in a stable repository, which means that it will be there stable forever. It will have a link that anyone can access at any point in time and would still be there. That is what it means. And we at Data in Brief have our suggestion of repositories that you as an author can access or can store your data in. So we do recommend a list. Elsevier also has its own list. But so you can feel free to upload your data in any of these repositories. But if not, if you want to use a different one, write to us and we can discuss this and figure out the correct repository for you. But yes, it is something that we take into consideration.
Okay, thank you very much. I would like to thank you for a very nice presentation, and I will pass now the floor to my co-moderator, Abdul Kabir.
All right, so thank you, everyone. I think it was really, really insightful lessons I was going through this webinar, and we thank you, the speakers, for taking your time to be here today. We really did appreciate your presence and your comments and your talks. It was really interesting. So, Jason.
All right. Thank you, everyone, for joining us today. And again, thank you, a very, very special thank you to our speakers and moderators, and of course, our ASPB members. As a reminder, you could watch the full webinar on the Plantae YouTube channel and all the materials that will be posted in the webinar and the Plantae blog once it is processed. Again, have a great rest of your week, and we hope to see you in our next webinar. Have a good day, everyone.
Thank
Bye.
you.